afternoon, everybody. Hope you can find a seat. It's my pleasure to introduce this Love Your Brain session. Happy to see so many faces and so many people attending this meeting. My name is Marion Leboyer. I'm professor of psychiatry at University of Paris in France. I'm also a very active member of the EC committee here at the ECNP and also the chair of the ECNP network in immunopsychiatry. So today I have the pleasure to introduce Professor Ilia Karatsoreos, who is chair at the Department of Psychology and Brain Science at the University of Massachusetts in the States. His research, as he's going to show you, is focused on the intersection of biological rhythm, stress, and immunology, and how this modulates resilience and vulnerability to environmental challenges. Thank you very much, and the floor is yours.
Thank you for that wonderful introduction, and thank you all for coming. I'm going to be sharing with you some of the work from my lab, but I'm also, in the spirit of this session, going to try to contextualize this in a sort of broader schema. I looked at the program, and I looked at previous versions of this, and I rapidly redid my talk this morning, so this will be a beta test for all of us. Let's see how it goes. I want to thank my lab for all the work that they have done in this, and I will highlight some of them along the way. I'd also like to thank the different funding agencies that have contributed to the continued support of my lab. I also have no other disclosures to make. The goal for today is for me to really try to explain the circadian clock's position in brain-body function. There's no way I can do that in 20 minutes. So I will be taking a selection of things from a couple different places. I want to discuss the impact of circadian disruption then on brain behavior and physiology. I want to start by just orienting us all for why biological clocks are so important. This is just to highlight that the biological clocks have evolved over billions of years. And in mammalian systems, our biological clock is about 500 million years old. It is incredibly highly conserved, and it really permeates every biological system. So in my lab, we tackle this by really taking a holistic approach and exploring the role of biological rhythms in many different areas, from stress biology to immunology and neuroimmunology and metabolism. And the way I like to think about this is that the circadian system is really critical in helping the brain to maintain balance through all these different biological systems in the periphery. And this is a cover of a review paper I wrote with my late postdoctoral mentor, Bruce McEwen. And the artist apparently read our paper, was so confused, she didn't really know what to do. And she threw this picture together, and she said, did I get it? And I said, it's absolutely perfect. This is really all about the way the brain maintains this balance and regulates constant changes. And I like to think about the clock as helping to constrain these different systems so that they operate within a certain set of parameters. They're able to adjust when they face a stress or a challenge, but then they have to come back down to normal function, so that we don't get into this situation of allostatic load and allostatic overload. So this is sort of the context in which I approach biological clocks. So if we look at timing, it really affects every kind of physiology and behavior you can find in a human being. So over here on the left is a cartoon. I like to joke to my students, this is me. So I get up at 6 a.m., I shower, I go to the lab, watch some TV, and then I'm back in bed by 6 p.m. And while all this is happening, there's all these different physiological rhythms that are going on. And Jürgen Aschoff, back in the first half of the 1900s, was able to measure many different kinds of behaviors and physiological outputs from graduate students that he locked into a bunker. So they were completely bereft of any outside timing information and was able to see everything from body temperature to cardiac output to oxygen consumption. Shows very clear daily rhythms. Since then, we've come to realize that there is a central clock that is located in the brain. It's in the base of the hypothalamus in a region called the suprachiasmatic nucleus that receives direct light information from the retina and then communicates this information through a variety of means, both neural and humoral, to oscillators all through the brain and also through peripheral organs. In my lab, we're primarily interested in brain oscillators, particularly those in the prefrontal cortex. We're also very interested in the liver clock and how it affects metabolism. And also these little guys here are the newest addition to the lab, which are microglia, and how they might be mediating some of the effects of biological timing on neuroimmune function. So because we see there is this really broad impact of circadian timing on physiology, you can imagine that disrupting clocks has widespread impacts as well. And every time I give a talk, I like to customize it for where I am. So what I'm showing you here is Milan from the space station. So this is taken back in 2015, just after the city government replaced the lighting in the city center with very bright LED lights. So you can see the city center very bright and the suburbs fundamentally different color because they stayed with the less blue non-LED lighting. What this is meant to highlight is the many ways, the many pervasive ways that our biological clocks can be disrupted. In this case, the impact of light at night. Many of us, myself included, because I just came yesterday evening, jet lag is another way we see impacts on biological clocks. So if we fly from Massachusetts to Milan, I have to adjust my clock. I have to adjust 6 hours ahead. I have to wake up early. I have to do what's called a phase advance. So this shifts my brain clock, and then all of my other oscillators throughout my entire brain and body have to adjust. Maybe next time ECMP will consider doing Honolulu for a little bit of a change. And if we go there, what we're going to have to do is we're going to have to wake up later and do a phase delay. So whenever we undergo jet lag, we're shifting these clocks, and everyone can attest to the way that you feel after a single incident of jet lag. So you can imagine the cumulative damage this has for individuals who live constantly out of time. We can also get jet lag without traveling, and I wouldn't be a card-carrying circadian biologist if I didn't use this opportunity to tell you all why we have to get rid of daylight savings time and stop the time shifting. What I'm showing you here is the number of car accidents, and over here on the right, the incidence of stroke before and after the time shifts, either in the spring, or the open ones over here, or the fall, the black ones here. And hopefully you can see that after our time shift, we have pretty big changes in both car accidents and cardiac events like stroke. And so this is from the Daily Mail. They put together a really nice little infographic on here. So we have a 24% increased risk of heart attack, a spike in depression, higher risk of stroke, and 6% rise in fatal car accidents, all from changing 1 hour. So this is just something for us to really be aware of. What I think this also does is it highlights that the circadian clock really does affect the entire body. It affects your brain, it affects the way the brain communicates with the periphery, and it affects the way those peripheral systems interact with each other. So in my lab, the question that we're currently asking is, how does time of day impact normal brain function? So if we want to understand how disruptions of the clock can lead to changes in behavior or physiology, it's good to understand how it functions under normal homeostatic conditions. I'm just going to focus on the prefrontal cortex now. There's lots of beautiful work from my group and many, many other labs focusing on other brain areas. I'm going to zoom in on the PFC in the interest of time. So the PFC is really critical for complex cognitive behaviors, decision-making, emotional responses, and it forms part of a much broader circuit, including the hippocampus, the amygdala, and many others. So it's really centrally focused. And one of the questions that my lab asks is, how does time of day actually affect the way this part of the brain works? One of the things we were interested in was, how does it affect metabolism at the level of cells and circuits? So for that, I'm just going to quickly introduce the concept of the lactate shuttle, which is one of the ways that neurons get a lot of their energy. So astrocytes help to feed neuronal function through the production and release of lactate, which the neurons then take up on an on-demand basis. And actually, this is one of the things that helps them function, particularly when meeting the needs of increased activity. And a very talented graduate student in my lab, Naomi Wallace, used implantable biosensors to be able to, in real time, track the amount of this extracellular lactate that was present in the prefrontal cortex. And hopefully what you can see here is that under normal control conditions, we have very beautiful rhythms of extracellular lactate in the prefrontal cortex. And importantly, this rhythm persists even when we put the animals into constant darkness, so they have no external cues. So what this tells us is that under normal baseline conditions, we have very nice daily rhythms of an energy substrate in the PFC. More recently, and again, I'm just really hitting the surface here, please take a look at the papers or ask me questions afterwards for more information. More recently, we've actually started to probe how the circadian clock fundamentally alters basic cellular properties, the basic properties of neurons that help them do all the different things that they're doing. And what I'm showing you here is work from a talented postdoc who's now got his own lab at the University of Wyoming. What he did was look throughout the day. Now, these are heroic experiments, just for everybody out there, means collecting animals at 4-hour intervals over the entire light-dark cycle, sitting at your ePhys rig at all hours of the day, trying to get as much data as humanly possible. What you can hopefully see here is that, particularly in males, this does not happen in females remarkably, and I'm happy to elaborate in another time, because females looks like a different part of the PFC circuit is rhythmic rather than these pyramidal neurons that we're recording from in layer 2-3. What we can see here is that in males, neurons are much more, these layer 2-3 pyramidal neurons are much more hyperpolarized during the dark or active phase, which then has significant impacts for how they actually process information. And what it looks like is that during this active phase, they have a much higher threshold for firing, and when they do fire, they fire at a decreased frequency in this active phase. And we're thinking about what this means for how this structure works, but you cannot deny now that at a very fundamental level, the biological clock is influencing this really fundamental level of neuronal activity. But let's get into the big question here is, what happens when good clocks go bad? What happens when these clocks that are affecting all levels of physiology are not working correctly? And in my lab, we use a model of environmental circadian desynchronization, in which what we do is we change the length of the entire day, which forces animals into a constant state of circadian desynchrony. So what I'm showing you here is a little cartoon. The light bar is the light, the dark is the dark phase, and these different colored lines are different underlying rhythms, whatever you want to call them, locomotor rhythms, feeding rhythms, you name it. And what we do is we compress the day to 10 hours of light and 10 hours of darkness. This is beyond the ability of the animals to synchronize to, but they're still affected nearly every day, and you can see them constantly moving back and forth, trying to adjust to this light-dark cycle. And what we find is this really pulls apart these different rhythms. The consequences seem rather profound. Back when I was a postdoc, what I showed was that this process actually impacts the ability of mice to cognitively change what they've learned in a Morse water maze. So when they do this Morse water maze task, they're learning the location of a platform, and what you can see in both cases, they learn the location of this hidden platform just fine. But then when we reverse the location of the platform and put it in the opposite quadrant, you see on the first day, everybody does pretty poorly. They don't know where I've moved the platform to. They have to relearn that. And then on the second day, while we see the control animals recover back to where they were before, our circadian desynchronized animals still struggle to learn the location of this platform. And it seems that they're doing this by making a lot of perseverative errors, and they keep going back to the old location over and over and over again, a hallmark of PFC dysfunction. This procedure also remarkably changes the structure of the neurons in the PFC. So after only 6 to 8 weeks of the circadian desynchronization, we see that the apical neurons, the apical dendrites of these pyramidal neurons in the PFC physically shrink. They become less complex, and we see them actually shorten. So this is showing us that we have these behavioral changes, we have these structural changes, and we've also now been taking this into the functional changes. So this is Naomi's data again here on the left with lactate in the PFC, and we can see that this energy metabolism is dramatically reduced in its rhythm now that we house them in the circadian desynchronized situation. So they lose the daily amplitude of their energy substrate, whether that's localized to astrocytes or an effect in neurons that then changes the way the astrocytes works. We don't know yet, but this is early days. Similarly, when we undergo the circadian desynchronization paradigm, the normal rhythm that we see in these PFC pyramidal neurons in terms of their membrane properties completely goes away. So we're able to completely change the way these neurons work at a biophysical level. So I'm just going to give you a couple of interim conclusions now, and I have a little more to talk about that's more on the immune side. But I want you to take away that normal brain function is impacted by the clock. This occurs at the level of neural structure, of function, and of metabolism. And disrupting the clock disrupts these processes. One of the questions that my lab is currently asking is, are these changes linked? So are we seeing changes that occur in energy metabolism that are then directly linked to the changes in the neuronal function? And then how does that relate to the changes in peripheral metabolism, which I'm not able to talk to you about today, given the amount of time, and how are these different processes linked? Let's just go back here to the general overall schema. And now what I want to focus on is some of the newer work from my lab over the past 10 years or so, exploring the link between biological rhythms and immunology, and biological rhythms and neuroimmunology in particular. So this is work that was done by graduate student Derek Phillips, who's now faculty at the University of Idaho. And what Derek did was ask, how does the circadian desynchronization paradigm change the way the brain responds to a peripheral immune challenge? So he challenged mice with lipopolysaccharides, which is a mimic of a bacterial infection, and gave that to them in their periphery. And then just 3 hours later, we explored what was happening in terms of their changes in their brain. And we saw in all cases, we see changes in the brain following LPS, but remarkably, the circadian disrupted group, this gray bar here, you can hopefully see, has a very different pattern of responses to this LPS. So in some cases, the response is very similar. So IL-1 beta in hippocampus, for instance, looks very similar to controls. But if we start looking at other responses like IL-6, we see a marked blunting of that response. In the hypothalamus, TNF-alpha is dramatically increased to the exact same challenge. So what we have is this fundamental disarray of the way that the immune system is responding, and then the way that the brain's immune system is responding to this peripheral challenge. Again, emphasizing the link between the brain and the body. Another student in my lab, Greg Pearson, took this one step further and asked, how does circadian disruption actually impact recovery following LPS challenge? So, as many of you in the room have experienced, you are almost always likely to get sick after traveling on an airplane. So you have your jet lag and you have your sickness. So you're in a really great mood. And how does that actually fundamentally change the way that you recover from this illness? So what Greg did was, again, using a peripheral LPS stimulation, he inoculated both control animals and circadian desynchronized animals with either PBS or LPS. What these raster plots are showing you, each one here, each line is an individual animal, and the little black ticks are, in this case, locomotor activity. And hopefully what you can see is that in controls, right after the inoculation, you see a dramatic decrease in their locomotor activity, as we would expect from sickness behavior, that rapidly comes back. But what we can see in our circadian desynchronized animals, this takes a lot longer to normalize. And we see the same thing whether we're looking at body weight, drinking behavior, feeding behavior, all the hallmarks of this type of sickness behavior. And we can quantify here again just on locomotor activity. What you can hopefully see, the blue and the orange lines in this case are LPS groups. The blue line is the LPS in the circadian desynchronized animals. It takes them almost double the amount of time to recover after this challenge with LPS. So what this is setting up is this idea that somehow the circadian desynchronization leads to this dysregulation of the immune system that impacts how both the peripheral immune system and the brain's immune system are actually functioning. We have new data from the lab that really complements nicely other work that's going on in several different groups, that microglia might be an important cell in this context. And it's kind of funny because when I start showing this slide, one of the first things people tell me, oh, microglia, they're so hot right now. It's the hottest cell. That's great that they're hot, but I went there because we really think they're actually playing a really important role in this process. So microglia are basically the tissue macrophages of the brain, and I'm sure many of you know about their roles underlying normal function. So how do they sculpt neuronal circuits during development and learning, their role in potential different neuropsychiatric disorders, neurodegenerative disorders, and also their response to injury and infection. And so we really think these microglia are playing an important role in many of these different settings. They have their own circadian clocks in them, so you can actually see that they oscillate. Lots of their different processes that are involved in sensing the environment and their ability to become motile and other aspects of microglia function are all under the control of the clock. And we have new data in the lab that I took out because I didn't have enough time to share it with you, is the response to an intranasal infection with a virus is really critical for survival, and that this critical survival role is different depending on the time of day in which we give the infection. So time here seems to play a really critical role in how the brain mounts an immune response, and we think that microglia are important in this. So I'm going to give you a few discussion points, and then I want to give you a couple of bright spots so we're not all down at how bad jet lag and circadian disruption is. Circadian rhythms are important in both health and disease. Understanding the role they play in health is critical if we're supposed to try to figure out how does that impact disease processes. Interfering in the systems that regulate these adaptive responses can compromise normal brain function and also change neuroimmune responses. So in many cases, one of the things that I think and many others think is happening with the biological clock is that it could set the stage for other insults and other things to have a bigger outsized effect on the brain when faced with challenge. So the broad reach of circadian timing means broad problems when they're disrupted. So you're not just affecting one system when you disrupt the clock, you're affecting basically every system. But I think that is tempered by the positive side of things, which means that the circadian system might be a really important way to actually get in and modify and intervene in some of these situations. So some bright spots here is trying to use light to improve mood and cognition. This is a really nice visual abstract from one of Samar Hattar's papers. And what it's showing us here is light acting through the retina, through two different kinds of retinal ganglion cells that are not involved in visual processing, but are intrinsically photosensitive, so they respond to light, but not in a visual sense. They send their signals to that brain clock, which then can impact lots of different kinds of hippocampal learning and other kinds of learning. But also it seems that a subset of them project to many other areas, including, in this case, he's highlighting the habenula, directly, suggesting that light from the environment can directly modify some of these brain areas that we know then are implicated in different mood disorders. So we have both mood and cognition being modulated by light. We know now also that light can be an effective treatment modality in the elderly, dealing with Alzheimer's disease and other neurodegenerative diseases. So it seems that this pathway can be a very powerful tool to help modify outcomes. So with that, I would like to thank you all for listening to my jet lag-induced ramblings. I hope there'll be some questions, and I will be around afterwards for more questions if we don't get to everything. So thank you very much.
So thank you very much for this extremely interesting talk, ready for mechanism, biomarkers, and treatments. So we have lots of questions, so I'm going to try to select some of them. Sure. If you want to sit here. So we have lots of questions on the link between all what you found and you showed and suggestions for people compelled to shift job. What would be your advice for them?
So there's lots of advice that's out there. The first thing I'm going to say is you have to try the different approaches to see what works for you. But one of the best pieces of advice which I did not follow today because I was still sleeping at 10 a.m., was to try to shift over to your new time as fast as possible by shifting your feeding schedules first, getting as much bright light in the morning as you possibly can, go for a walk, go for a jog, do these different things, and try to stimulate your circadian system as early as possible and as strongly as possible through as many modalities as possible. So feeding, we know, can help to entrain and synchronize biological clocks. Light does the same thing. So if you can give the double whammy of light and food and throw some exercise in there, it should really help.
We also have lots of questions related to light. So one question is, can you comment on infrared light and the importance of brain health? Another question on blue light. So any comments on that? Yeah, so this is a really active area of research. I'm not too sure about the infrared light. That's not an area that I'm particularly expert in. Blue light, though, is really a potentially powerful tool because those retinal ganglion cells that are intrinsically photosensitive are skewed to respond more to the blue light wavelengths. So blue light and blue enriched light has a much larger impact on those cells than just a regular white light that's not blue enriched. This is why, for instance, many of you have night mode probably activated on your smart devices and on your tablets and on your computers. And what you see that does is it shifts it to the more orange part of the spectrum. There's not a heck of a lot of work that's actually been done that I've seen that's been really well controlled, but reducing that amount of blue light, blue enriched light late at night, for example, can very, very much help you get better night's sleep and cause less disruption. On the treatment side of things, blue light blocking glasses have been used in the treatment of bipolar disorder. They've been used in elderly individuals, so to reduce the amount of blue light that they're getting at some times of day, and then helping to, thereby helping to increase the amount of blue light that they're getting at the better time of day to help sort of consolidate and shift their clocks to keep them more consolidated and less dysregulated. So light is incredibly pervasive, as we all know, and it sneaks into every aspect of our lives. And so finding ways to minimize exposure to light, particularly blue light, late at night, is definitely something that we should all strive for. So we have no time for the last question, but I can't resist. So can you comment on the role of circadian disruption in neuropsychiatric illness? And we'll stop there.
Yeah, so this is, well, thank you very much for that question. And I'm not going to tell you it's not complicated, but it's very complicated. The question is sort of a chicken and egg one, I think. So in some ways, I think that circadian desynchronization is a risk factor for leading to other different kinds of psychiatric disorders. Why? I think because of some of the things that I showed you today. It changes the way the neurons work, the way the neurons work with each other, the way they get information and are able to process that information, the way that they're able to recover from any kind of insult or injury. At the same time, many of these disorders cause changes in brain structures, including the suprachiasmatic nucleus, that might then lead to changes in sleep patterns, locomotor activity, and at that point could be a really good diagnostic indicator for, I think, some of these things. So I think it's really, it is a loop, and where we get into the loop to help with interventions, I think that's the multi-million-dollar question.
So we'll leave it with that. Thank you very, very much. Thank you for your coming, and have a great day.